Good afternoon. My name is Ronnie Green. I have the pleasure of serving as the Vice Chancellor of the Institute of Ag and Natural Resources here at the University of Nebraska-Lincoln. Welcome to those of you here in Hardin Hall on East Campus with us. We have a capacity audience today. It's really great to see. And I've been directed to tell you that if we're out of seats, that there are overflow rooms available where you can watch the lecture in this same facility as well. We are very pleased to welcome you to the inaugural lecture for this academic year of the Heuermann Lectures. And just a bit of information for you about the Heuermann Lectures and what they represent and stand for. This is our third year in this lecture series. We were able to establish it due to the generosity of the Heuermann family. Keith and Norma Heuermann in particular have helped us to bring these lectures to our campus, where we invite international authorities on topics around food security, natural resource security, and rural landscapes to our campus. We're very pleased today to have Keith with us. I think he's been at almost every one, has been at all of these lectures. Keith, if you would stand and be recognized, please join me in thanking Keith Heuermann. And we also are pleased today that Keith has some family members with us, and I know they probably don't want to be pointed out, but his sister Alma is with us and her husband Norman, so thank you for being here today as well. This year's inaugural lecture is on a topic that certainly has been receiving a lot of attention, not just in the most recent past, but over the last several years, around GMOs and GMO food. And we are particularly pleased to welcome one of our own to the lecture stage today, Dr. Sally Mackenzie, who is a professor jointly in the Department of Agronomy and Horticulture and the School of Biological Sciences here at the University of Nebraska-Lincoln. Dr. Mackenzie was the founding director of the Center for Plant Science Innovation here on our campus, where she worked with a dedicated and a talented group of faculty working in technology that often leads to the ability for us to have genetically modified plants. Her research focus areas are in the areas of plant genetics, epigenetics, genomics, and organelle biology, organelles in the cell that are responsible for its metabolism and function. She received the Sigma Xi Outstanding Scientist Award here at UNL in 2010 and is an elected fellow of both the American Society of Plant Biologists and AAAS, or the American Association for the Advancement of Science. Sally has served as associate editor of the Crop Science Journal and on the editorial board of the Journal of Plant Molecular Biology, Journal of Heredity, and Plant Physiology, as well as on the advisory editorial board for Plant Journal. She serves on the executive committee and chairs the publications committee of the American Society of Plant Biology, and was instrumental in that group coming forward with a plan for the future of plant research and plant genetics research just recently. She's the author of numerous referee journal articles and invited book chapters. She holds 3 patents and is an invited lecturer nationally and internationally, and in particular on the topic that you'll hear from her today. So please join me in welcoming to the Heuermann Lecture stage Dr. Sally Mackenzie. Her title today is Beyond GMOs to a More Honest Dialogue About Our Food. Please welcome Dr. Sally Mackenzie.
Thank you very much for this wonderful opportunity. And hopefully, I'm on. You know, I received a degree in plant biology from the University of California, Davis many years ago, but my experience with agriculture didn't start then. My experience with agriculture began when I was 4 years old. So I'll tell you that I do remember the good old days of agriculture before GM technologies, because I had a father who worked in fresh market agriculture in California. And so I can remember many an early morning standing out in his tomato fields or strawberry fields or any field and having one of these crop dusters come over my head. And I remember at the time it felt like fairy dust. I actually enjoyed the process and it would sort of tingle on my skin. But of course now, as an adult, I every year have my full panel of liver enzymes assayed. And yes, I did lose my mother to liver disease, and we will never know why. So those are the old days for agriculture, I hope, because there are better and safer ways to do agriculture. But before I talk about agriculture, I want to talk a little bit about the way we think when we're presented with new concepts. And I can tell you, at least from my perspective, that we're going to have to get used to some of these new concepts because the world has changed so much. So here I want to talk to you a little bit about one of the problems that does come up in my home state of California, where I grew up, and that's the problem, of course, of water. We never had enough when I was growing up, and they have even less now. And it is one of our major agricultural states. So of course, several of the civil engineers came up with the ingenious concept of recycling water. It's what we must do, they said, especially in the urbanized areas of California around LA and San Diego. And of course, to recycle water is a very new idea, and it involves very new technologies. But these civil engineers were sure they could make it scalable and that they could make it economic. And of course, there was the response. That didn't feel right. In fact, the whole concept made Californians a little queasy because, of course, it wasn't natural. It didn't matter that the water that would come out of that system would be absolutely more pure than the water that was coming out of the ground to begin with. That didn't matter. It was a concept. It was an impression. It was up here. A lot of what we think about when we think about genetic modifications of crops is up here. It's not about the science. The science has already been done, just like the science has been done on water systems. But we don't feel right about it. There's a lot in common between these two. The Clean Water Action Group in California accepts water recycling, but wants it to be tightly regulated. No matter what the scientists would say about this water system, the public thinks that directly reusing sewer water is plain gross. And toilet to tap is a term used by opponents to disparage the system of producing safe and pure water. A lot of parallels with what we deal with when we talk about genetic modifications of crops. It's a natural reaction. And so we can learn something, I think, we scientists who are in this business, from this example, because California's pretty much getting close to being over this hurdle. The scientists dealing with the water issues in California came to the conclusion that we have to think about this in a very different way as we approach it. So what they did is they took several psychologists and they went out and they started to do polls of Californians. What the problem is, what would it take, what would feel natural to you? And it turned out that in the end, if we took that absolutely pure water, once it had been purified, and we put it back into the system from which we think it came. We either pumped it back into the ground or we put it into reservoirs. Now, of course, we're recontaminating this, but that's not the issue. It felt more natural that way. So you're looking at an absolutely pure water now in this reservoir in California. And yes, there's bird poop falling in it and there are insects falling in it, but we feel better about it because it's more natural this way. And of course, now this water can be used, putting it back into the ground. But the more important issue wasn't that they found a compromise that people were comfortable with. It was that the conclusion was actually that between climate change, increased urbanization, and growing demands for food and energy, there's no way around it. It is inevitable. It is part of our future. And thus we need to start an honest discussion about what is going to be our future, because the fact is that we have these very same issues confronting agriculture. So I first want to start with this concept of unnatural because it's the first thing I hear whenever we talk about genetic modifications. We talk about recombinant DNA as being unnatural. So I hope to convince you first that in fact it is absolutely natural, because in fact recombinant DNA, as every geneticist knows, is the biological driving force of nature. It is what Mother Nature strives for. If I were giving you a class in genetics, rather than just telling you about genetic modifications in agriculture, I'd start with a diagram like this. And I would remind you that recombination, DNA from different sources coming together and exchanging, is the fact of life. It is why you have your mother's blue eyes and your father's dark hair, and it is why we have so much diversity in this world, and that is the driving force of evolution. And I would tell you that the blue comes from dad and the red comes from mom, and DNA tends to be nicked easily so that it will have to repair itself, and in that repair process it will seek new DNA sequences. And as a consequence of all of that, you will have new sequences hemmed together in order to create diversity, because diversity is the stuff of life. You know, let me just remind you that the more diversity we have, the more opportunity for survival. This is the way this works. But what you never comes across in the genetics class that I used to teach is the understanding that recombinant DNA and DNA recombination is everywhere. It isn't just in those examples that we understand. It is in bacteria that have no problem with interspecific DNA exchanges. And you know that when you walk into any hospital and you learn about antibiotic resistance, and one microbe figures out how to do it through mutation, and then sends it on its way throughout whole hosts of microbes that absorb that. And it doesn't matter the species. They're happy to share that information with everyone, which is why we have to worry about overuse of antibiotics. Happens in fungi as well, and in fact there's a new fungal infection in the Pacific Northwest affecting humans that comes basically from recombination exchanges of DNA in fungi that we were not intending. And of course, recombination of DNA is the underpinning of plant breeding. It is what we strive for, is to maximize diversity and to find ways to create new combinations so that we can come up with the crops that we're going to need. And of course, it happens naturally. So here you're looking at a coyote-wolf hybrid that happens in nature. I didn't know these happened. I just learned about these not too long ago. I think they're adorable myself, but they happen naturally, and they happen because nature has this driving force to come up with very, very new combinations. Well, you might think all of these are still in your realm of natural. So let's put it a little further out there. Here you've got a daughter, a parasite of plants, not even remotely related to the tomato plant that it's infecting there, and it's sticking its haustoria down into that surface of that leaf or of that stem, and basically sucking out nutrients. That's what parasites do. But of course, while it's sucking those nutrients, it's donating its DNA to the genome of that plant. And likewise, that host plant is donating its DNA to that daughter. That exchange doesn't feel so natural anymore. Those are very different species. Let's take it one step further. Gauldiaria is an alga. It's a plant, a lower plant. And it lives in very extreme environments, high in metal toxicity, very high in temperature, very high in acidity, not the kind of places that plants like to live. So the phenomenally wonderful way that it has learned to adapt to live there is that it has loaded up 5% of its genome at least with sequences that it naturally acquired from bacterial genomes that gave it its capability to live in this very difficult environment. Basically genetic engineering, naturally. Bacterial sequence is transferred right into that algal species that allows it to live in an adverse environment. Who knew? And in fact, nobody knew. This just came out in March 2013 in the journal Science. Lovely story. Or the deltoid rotifers. Many of you remember in your high school biology class when they would go and they would get that drop of pond water and put it under the microscope so you could watch everything swimming around. Those rotifers basically have had only females in their population for about 1 million years. And females all by themselves don't do so well at recombination. But that's all they've got. And so they reproduce parthenogenically. And that's a bad deal, because that means that when a rotifer gives rise to another rotifer, gives rise to another rotifer, they're all identical. No diversity. So what did Mother Nature come up and do here for genetically engineering that diversity? Those rotifers go into a dormant stage when the environment gets adverse. And when they come out of that dormant stage, they absorb basically any DNA around them. Plant, fungal, bacterial, you name it. And that is the stuff of the diversity in rotifers. How lovely an example of natural genetic engineering. But let me share with you the natural recombinant DNA process for plants. This has to do with what you're looking at here, which is crown gall on the stem of a plant. And that crown gall actually emerges as a consequence of the fact that there are bacteria that infect plants. And when that bacterium infects the plant, it injects its own DNA into the plant to basically reprogram the way those cells behave. And when it introduces that foreign DNA, the plant cell absorbs it and changes its behavior. Genetic engineering, bacterial DNA into plant. It's quite natural. But in fact, it is this system that biologists have learned from. Agrobacterium tumefaciens introduces its own bacterial DNA. Why can't we introduce a gene of our liking into that bacterial DNA so that it will do all the work for us? And this is basically at the heart of GM technologies as we know them today. We've simply taken advantage of a very natural process that goes on in the wild all the time. We've simply exploited it for a plant breeder's desire to have new traits that we cannot find in nature. Now, some of those new traits we can't find in nature because we don't have enough diversity left. Too many plants have gone extinct. We don't have enough diversity to be able to come up with what we desire. And part of it is because we have created a very unnatural, very intensive agricultural system, namely monoculture, and growing acre upon acre of corn requires certain environmental capabilities that those plants don't come naturally equipped for. But if you want truly natural, and you don't want man to be really tinkering with the genetics of a crop, then feel free to enjoy your carrots, because that's what you get. This is man-made. Feel free to enjoy a cob of corn, because this is man-made. Basically, feel free to enjoy your natural tomatoes, but don't eat all of them, because the alkaloid levels are enough to kill you relative to the tomatoes that you buy in Hy-Vee regularly. This is what is man-made. This is the huge accomplishment that we've been relying on for 1,000 years. Every plant breeder in this room and every plant breeder in this country has to feel extremely deeply proud of what they have done in feeding the world, because that's where it has come from. You cannot eat that corn until a breeder works the magic that breeder works. So when we talk about the new age of plant breeding, we need to understand that we've been relying on the plant breeder for 1,000 years. Now, when we talk about organic as being really natural, then I want to basically burst your bubble just a little bit. Because that beautiful ruby red grapefruit that we eat, labeled organic, that beautiful ruby red color came because some breeder discovered that if you gamma irradiate crops, it will induce mutations. Most of them are mutations we don't want, so we don't use it a lot in our plant breeding. But some of the mutations turn out to be quite nice. So that ruby red color that you enjoy in your organic grapefruit basically came from gamma irradiating it. And the problem that I see with that, not too many, but one of the problems is that when a breeder went in and did that, yes, we came up with the ruby red color, but we probably came up with 50 more mutations that we don't even know about. And we couldn't have predicted, and we don't know the consequences of. Likewise with those beautiful colored carrots, labeled organic. Mutation breeding. We also use chemical mutagens sometimes to introduce these mutations. Does that feel natural to you? Because it's called conventional plant breeding. I don't see it as any less natural than what we're doing in GM. The only difference with genetic modifications using agrobacterium and GM technologies is that we know precisely what we're doing, we know right where that sequence goes, and we know exactly where it's expressed. It's just a more precise way of doing virtually the same thing. And then, of course, there is this issue of what we're actually doing with GM. And you will hear often, and I had that scrolling of many of the things that you've heard often or read on internet, you'll hear often that the things that we're putting into GM crops are allergenic. And especially, I don't know why, BT corn really gets beaten up over this. BT corn is a corn that has been developed to be insect tolerant by virtue of the fact that it expresses a bacterial gene from Bacillus thuringiensis that actually gives it the capability to be an insecticide. Now, insecticide sounds dangerous, but this is a protein, so it winds up getting digested in your digestive tract if you get any of it. And what you eat is minute amounts. So people feel fearful of this. This protein seems foreign, and this protein seems insecticidal. That doesn't sound like a good thing, therefore it must be dangerous. But where that technology came from was more than 100 years ago, when French farmers started to spray Bacillus thuringiensis, whole bacteria, on their crops. And as a consequence of spraying large amounts of this bacteria on the crops, they found that it was a natural insecticide, a biological insecticide. This is the bag that you buy it in. Now, of course, there are no regulations on how much of that BT, basically the cry protein, that BT toxin, is remaining on those organic crops that we grow, or those organic vegetables that we grow. We have no idea how much is there. We know precisely how much is expressed in BT corn. So if this is the allergenic protein, then I would submit to you that those allergies are likely arising from eating organic produce, not from eating BT corn. The vast majority of BT corn you're eating, you're ingesting after the cow has eaten it. But the vast majority of the BT that you're likely consuming in organic is sitting as residue on the surface. Now, I'd expect most of that residue to dissipate and degrade, and certainly this is a protein. But I just want to point out to you that when we talk about regulations, you need to think clearly about overall regulations of our agricultural system, and you need to think clearly about exactly what these proteins are that we're working with. Because for 100 years, the French were finding this protein to be no problem at all, until we engineered it, okay? So basically, I'm hoping that I'm alleviating some of your concerns about what scientists do, because scientists give a lot of thought to exactly what's been tried before it's ever been tried in a transgene. So one of those things I had rolling there was an article entitled "Discovery of BT Insecticide in Human Blood Proves GMO Toxin a Threat to Human Health." Well, A, I don't believe it's in human blood, but if it were, I'm more concerned about it getting there from our organic produce. And I'm not advocating that we now regulate BT toxin, I'm just pointing out to you the realities of this argument. Because when we think about food safety, we have to think a little bit about how we assess food safety. And there has to be a little bit of a rational thought there, because you're always trying to prove a negative. You'll never prove that anything is absolutely safe. Do we feel that our meat is safe? Yes, we eat it every day. We in Nebraska especially. But we all know that there have been serious dangers associated with our meat supply, particularly with E. coli. And our food science department here works mightily to make sure that that never happens again. There's a lot of research that goes into that accident. Likewise, we know that eggs are safe. Most of us eat them most mornings. But we also know about the hazard of salmonella that we've encountered in the past. We know that our organic produce is generally safe, but we also know about the hazards we've had in the past. Let me point out, most of those food safety hazards dealt with microbes. And so it is important that we have enough understanding of the microbial populations that inhabit our world with us, those that are safe, those that are not. And I would urge you all to consider taking a microbiology class. But that has nothing to do with GM technology. Historically, we've learned through experience what foods are safe to eat. Those that have to be processed, those that have to be avoided. So we know that wheat causes celiac disease in some people. It doesn't have anything to do with GM technology. And yes, possibly GM technology will help address this problem if wheat will ever allow GM technology. And of course, legumes have to be cooked to destroy lectins and trypsin inhibitors. They'll make you very sick if you don't do that. We've learned that. We live by that. Is that food unsafe? No. You've learned how to consume it. We know that most foods are allergenic to some people. I've got friends with allergies to strawberries. Does that mean strawberries are unsafe? I've got a lot of friends with allergies to peanuts, and I wish the GM technology would actually address that problem, because that can be really hazardous to the health of people who are allergic. But does that make peanuts unsafe? Basically, no. We accept those hazards, we accept those risks because we understand them. And so therefore, the assessment methods that we use must be scientifically sound, but they must also be relative. And that's why the FDA states that GM crops should be as safe as the same type of conventionally bred crops. And in fact, they've gone well beyond. We have no health issues that have arisen yet for GM crops, and we hope we never will. So where did GM technology come from, and how is it that Monsanto gets full credit for everything? This has really perplexed me because Monsanto is just in the headlines of every anti-GMO website I could find, and I cannot figure out why they think Monsanto is behind this technology. In fact, in '82, the first genetically modified plant came from scientists in research centers. It was an antibiotic-resistant tobacco. And in '86, the first field trials were done, both in the US and France, on an herbicide-resistant tobacco. Most of that was developed in Europe. And by '87, the first company to develop Bt tobacco was not Monsanto. It was Plant Genetic Systems that was headed by two renowned scientists in Europe, one a Belgian, Mark van Montague, and one a German, the late Jeff Schell. They were both researchers. 1992, China, not the US, was the first country to allow commercial transgenic crops. Okay, virus-resistant tobacco was the crop of interest there. And by '94, Europe was the second. That was the first GM crop to be marketed in Europe, which was an herbicide-resistant tobacco. I haven't mentioned Monsanto yet. Only by '95, when we had the first Bt crop, that was in potato, that the EPA approved in the US. Canola, basically to make modified oil composition, Calgene took that on. Bt corn, Ciba-Geigy. Herbicide-resistant cotton, Calgene. Bt cotton, Monsanto. In swift competition with Calgene, they eventually absorbed Calgene. Glyphosate-resistant soybeans, virus-resistant squash, delayed ripening tomatoes, that was when everything came out. Monsanto was one of many. By '96, we had 35 approvals to commercially grow 8 transgenic crops, 1 flower crop, with 8 traits in 6 countries, not just the US. So I just want to correct a little history there. Monsanto really didn't do all of this. It actually comes from mostly Monsanto's competitors, who you might notice never come to the defense of Monsanto when they're in the limelight. And by 2000, golden rice. And for me, that was the start of a golden era, because finally we were thinking about something other than profit. Golden rice for me was such a statement for the scientific community. And I am so proud of Ingo Patrikas for wanting to do this, wanting to make a difference. This is why scientists got into science. It was not at all about profit. It was about making a difference and feeding the world. And Ingo Patrikas went for it. So what has been accomplished? Well, you know, I think that a lot of biologists like to brag about the fact that as much as we have not really appreciated GM, our consumers have not appreciated it, the fact is that a $45 million industry in Hawaii was saved because someone was clever enough, someone from Cornell University was clever enough to know how to engineer virus resistance at just the right time. It was a big deal at the time. It saved the industry. And of course, now we see plumpox virus. It hits peaches, it hits plums, it hits cherries. And basically now, again, we've come up with a C5 construct, and this turns out to be the most devastating disease of stone fruit, but now we're not so sure that we want to deregulate this product. We're not sure we want to do this again. Now, of course, you must all know about Roundup Ready soybeans, and there's a certain controversy associated with those, and Yield Guard corn, which of course has that BT trait I was talking about earlier. In all cases, we've seen increased yields. People will say we haven't, but we have. And it defies logic that you wouldn't. If you can control weeds and you can control insects, you know that you can improve your yields. And yes, we do have lower inputs. We're not tilling as much. And yes, we do have lower costs, fewer insecticides, fewer spray, or a less rigorous spray regime. We have reduced erosion. All of these are good. And the additional payoff, we don't have so many fungal toxins because we don't have so many fungi being brought in with those insects to invade our crop after we harvest it. All good things. Mostly what we're hearing about now are the not so good things, and they are true as well. When you like a good thing too much, whether it's genetically modified or whether it's conventionally bred, if you like an herbicide too much because it is so safe to apply and so effective, you will use it all the time, and it is no different than using an antibiotic. If you use it again and again on the same fields, you will eventually see those weeds develop mutations that allow them to be tolerant. It's just the way of Mother Nature. We've known it, and we have not managed, or not all growers have managed their crops properly, and so therefore we are suffering the consequences. Likewise, the same thing happens with Yield Guard in corn. Now, all growers are instructed by whoever is selling them this seed that they have to have a certain amount that is not Yield Guard in order to allow those insects to grow. But, you know, that's all wonderful space, and we'd like to have a little more yield this year, and so we haven't managed it properly. And yes, we are starting to see very low frequencies, but we're starting to see insects that are developing tolerance. So, this is the downside. This is the agricultural downside. And is it Monsanto's fault? I don't know, I'll let you be the judge. But too much of a good thing is always going to have a downside. That's all I'll say. But then it brings us to golden rice, where the World Health Organization estimates, you know, as high as 500,000, and in some parts, maybe 1 million, children become blind each year. And remember, vitamin A isn't just for your eyes, it's for your immune system. So a lot of these children die. So this is an issue. And so this was conceived basically as a public good project. This was part of the Golden Rice Humanitarian Board that was established. And the funding that went into this was not big ag. In fact, the funding went from the Rockefeller Foundation, the Bill and Melinda Gates Foundation, USAID, the Philippine Department of Agriculture, Harvest Plus. This was not a big ag project, never conceived to be a big ag project. And the beauty is that it worked. First time around, the vitamin A levels weren't so great. But by the time it was re-engineered, the vitamin A levels are superb in this line. And remarkably, those vitamin A levels were raised by Syngenta Foundation, They contributed the technology free of charge. Likewise, Monsanto contributed the IP for this free of charge. This was a humanitarian effort by all involved, and it just makes me proud to be part of this community. This is what it's all about, because this rice was conceived to feed the developing countries where it would really have and do the most good. As you can see, it's bright, bright yellow. There's a lot of vitamin A in this. So there has been the response that this is unneeded. Why don't we just have people eat carrots, eat sweet potatoes? And of course, if that were feasible in Bangladesh, that's probably the agricultural approach that would have been taken. But as you can see, the vast majority, here what you're seeing in black, of what is eaten, particularly in rural areas of Bangladesh, is rice. So if you want to impact the vast majority of the hungriest and the most malnourished people in Bangladesh, you're not going to be distributing carrots. You're going to be feeding them rice, nutritionally enhanced rice, and you're going to have a major impact. So in a few minutes, I'm going to tell you what's actually come of that big impact. But first, I want to just remind you that this issue about GM is, as far as I'm concerned, a distraction. The science has been done. You've all heard about the science. You've all been eating the product. We know, so far, we've not encountered any health effects. So this food supply appears to be the safest in the world. But the distraction comes in that what we should really be talking about today isn't that. It's about the fact that, especially you up there, you students, this generation that you're going to be in charge of, this new world you're going to be in charge of, has climate change, whether you believe it or not. And it has a depletion of arable land on which we're going to be able to do the kind of agriculture we've done in the past. So monoculture is going to have some real limitations. And we're going to see some real changes in the inputs, because you know what? We're running out of phosphate, and it's not renewable. And we're not sure what to do about it. And by the way, we're running out of water. And I gave you one solution, that which most people consider pretty gross. That's only going to be for urban areas. We're going to have to come up with some really clever ideas for how to make our crops not so thirsty. And all of those challenges are your challenges. And we don't know how to solve them, but we're going to have to come up with something by 2050, because we're going to have 2 billion more people to feed. So we're either going to have lots of war and lots of fighting across continents, or we're going to come up with some really technologically advanced ways to do agriculture. That's what we should be talking about, and I would submit to you that GM should be one of the tools in that toolbox, just one. So, at this point, part of the reason that I agreed to give this talk, which is a talk you know many of my colleagues don't give and don't like to give, it's not because there's anything to hide. It's because the science is already done. So most scientists stand up here and we don't know quite what to say beyond the fact that the science is already done. This has now become much more of a sociological and a psychological discussion. But the reason I'm here is that I read this article online in August this year. Is it time for scientist activism against GMO fear-mongering? And I decided maybe. And part of the reason I decided maybe were several events that were happening simultaneously with this coming out. And one of them was an article that came out this last year by Seralini, an Italian researcher, and his group. And what they did was they presented the results of feeding rats Roundup Ready corn. Some of these rats have been fed just Roundup Ready corn. That terrible looking rat in the middle was fed Roundup Ready corn and Roundup. And that really unhappy one on the right was fed only Roundup in its water. And these terrible, terrible things have happened. What Seralini and his group don't tell you is that basically they omitted the photos of the tumors that looked very much like this in their control rats that were fed non-GM. They were there also. They also had way too few rats for a study of this type. They let the study go for 2 years. And as a consequence, it was really statistically very difficult to tell whether there was a difference between those rats that were fed non-GM corn and those rats that were fed GM corn. They provided no report on the feed composition in that paper. They provided no report on the mycotoxins that might have existed in that feed. They declined to provide their full data set to the journal to which they submitted this after it was published, and there was an outroar by the scientific community. They declined to show their full data set to the European Food Safety Authority. And that's just not the way we do science, especially controversial science like this. Remarkable results demand remarkable demonstration. They also didn't really mention, although you can know it if you know rat biology, that the rats that they used are of a strain that was developed specifically to be hypersensitive to their environment and to produce tumors. This is a tumor-forming, spontaneous endocrine tumor-forming strain of Sprague-Drawley rats. The reason these were bred this way is to be more or less like an Ames test. They are hypersensitive to their environment, so like the Ames test, which was if you remember that back in the days when it was a bacterial plating, where you would extract extracts from broccoli and from various chemicals that are in our environment, and you would ask what impact that had on mutagenesis of those bacteria. And we could learn a little bit about cancer-causing agents. And in fact, broccoli had a whole bunch, by the way. Well, it turns out, in this case, these rats are somewhat like this. That's why it's so critical that you have plenty of large enough sampling, especially if you'll allow them to go 2 years, because they were never designed to go on these studies for 2 years, only about 90 days. So after that 2 years, they're all forming these tumors. And in fact, even sitting and rattling their cages over that period of time is enough to stress them out to likely form these tumors. This is the difficulty when you put out really exciting research like this, is that they forgot to tell you a little about what was in the fine print. And so there are a lot in the scientific community that were really in an uproar about this study. But then this summer, my daughters and I were traveling, and they love to read Elle magazine. I don't buy it very often because it's very expensive, but we bought this one. And as we're thumbing through, I see in Elle magazine that there's an article there called The Bad Seed. And as you scroll down, or I did with my eyes, I suddenly see that GMO is the topic here. This is a journalist who's going to tell you about the very terrible illnesses that she has suffered. And she and her doctor have decided it's due to eating BT corn. And of course, never tells you all the rest of what I told you. In fact, this can't possibly be due to BT corn, and she never had any allergy tests done. But of course, it made such a stir that she now has the opportunity to interview lots of scientists that reacted so that she can write yet another article. This is a little bit about the sensationalism. And I was angered by this because I felt that it was manipulating the public. Who do they suppose is reading Elle magazine but public who basically is not necessarily going to know what BT corn is and whether it can do this kind of damage? I felt it was wrong, and I felt that it was basically bad science. And then whatever happened to the promise of golden rice? And this is where I felt a little like activism was called for. So, as you know, any new product that comes out, people in general say it needs to be tested for safety. And if it's going to be tested for safety, we need to go out and we need to plant field trials and do real honest-to-goodness research to learn whether it can be grown properly and whether it can be consumed safely. So that's exactly what the International Rice Research Institute did with golden rice. Before they wanted to introduce it to the Philippine public, they wanted to grow it and see whether it was safe and whether it could be cultivated and whether it was acceptable to people there. All the tests that Greenpeace argues for. And instead, what happened when they put those plots out, as you can see, there are fences around them, is that Greenpeace bussed in 400 Philippine farmers and about 40 to 50 activists. And the activists, posing as the farmers, went in and trampled all of that research material, basically destroyed it in one afternoon. The group that they were working with, Mazapag, calls themselves a farmer-led network of people's organizations, but basically what it is it's an anti-GMO group, okay? And that's fair enough. They teamed up basically with Greenpeace on this project as well as on a project to secure a court injunction against GM eggplant that was designed to reduce insecticide use on eggplant, also for developing countries. And I don't think this is right. I think that if they're going to ask for testing, then they should allow the test to go on. And this was really an unfortunate event here. And of course, what they said they were doing was fighting against exploitation by huge ag corporations. But what I explained to you earlier is there are no ag corporations involved in this product, purely humanitarian. So let's talk about huge corporations, because it's important that we know who we're dealing with. This is Greenpeace. This is a Greenpeace activist who's illegally destroying a wheat crop that's being tested for GM in Australia. But what I learned in the process of studying a little bit more about Greenpeace is that Greenpeace itself is a huge corporation. So Greenpeace International worldwide brings in $335 million in annual revenues to fight against GMOs and to do much of their other work. They spend at least $189 to $200 million in this communications effort that Greenpeace makes. What you have and what we are dealing with when you wonder why scientists don't have enough of a voice in this argument is you have ideology combined with a vast financial resource. And what I find so frightening about this is the fact that when you combine money that way with such ideology, you have the ability to derail progress on all of the issues that we care very much about for the next generation. This is the problem that we've got, not a science problem. If we think about how production is going to be increased, it's going to take technology, changes in irrigation, changes in fertilizer, changes in pesticides and herbicide use, more land, which we don't have. We're going to have to develop precision agriculture systems. But what I want you to know is we also need genetics. We'll need to be able to make interspecific crosses. We'll need to be able to use chemical and radiation mutagenesis. We'll have specific genes inserted using transgenic technologies. All of that together is going to have to be part of this toolbox. And what comes of that, we hope, will be some new oil compositions. Those are some of the things that are coming from this university itself. We have new uses for some of the products that we have now in aquaculture to make it a more sustainable system. We have new herbicide tolerances that will allow growers to now possibly cycle away from glyphosate so that we won't have the problems we have with super weeds. We have potential GM solutions for citrus greening. Realize that the New York Times just presented the fact that we are about to lose our citrus industry if the GM technologies cannot solve this problem. It's a huge problem, and the citrus industry, important to California and Florida, is not likely to survive it using conventional methods. We're now looking at enhancing flavor and nutritional components, new disease resistances, and new approaches to yield enhancement. And that's what I want to close with, is that yield enhancement. And the reason I want to close with that is because all of these are basically single gene traits, except that last one. And yield enhancement is going to be the thing we're going to care the most about in the future. It's complex, we don't understand it genetically, but we're going to need it. Up to now, we've often used hybrids for yield enhancement, and they work great for the crops in which it works. But the reason I want to present this system is because it's important to the point I made initially of finding a compromise between those who oppose GM altogether and those who see GM but don't want it to take over all of our agricultural system. This was a discovery made by a research lab where they found that it was possible to downregulate one single gene in the plant, and by so doing, change the development of the plant altogether. The development of the plant now becomes, what you're looking at the top is on the left, you're seeing the wild type plants. This was the first plant they did this in. On the right, you're seeing those small dwarf plants. That's what they look like once they lose this one gene. But what's interesting is that this plant takes on all sorts of new forms, as you can see down below. Suddenly it grows like a perennial. It starts to get woody. It behaves like it's a juvenile plant for very long periods, never turning into a properly matured plant. All these unusual developments, delayed flowering, extended juvenility, this dwarf phenotype, they don't sound like agriculturally valuable traits, and they're not. But what this actually does is to reprogram the plant, to make it grow in a totally new way as a consequence of one gene. But here's the important part. We can move this into crops by downregulating, using one transgene, downregulating that same gene in all these crops, and the same effect happens. You see this dwarfing, you see this strange branching pattern, you see this funny looking tobacco plant over on the right. Again, not agronomically valuable, but definitely reprogrammed. And the really important thing is that after you remove the transgene, after you take it back out, so it's no longer transgenic, these effects remain. These effects are called epigenetic effects. They're actually changes in the way that genes are expressed, but you haven't changed the plant genetically. You put the transgene in, you take it back out, but you have these effects. Now, why do we care about this? Well, one is that it's a heritable effect. You can go 10, 15, 20 generations, and these same dwarfings remain stable. Well, remember, we still don't care about dwarf plants, and that's not the angle that we want for our crops. But this effect remains stably, non-transgenically. And the most important part is that if you take one of those altered plants, like you see on the right here, strange in its branching and weird in its tillering, and you cross it back to the original plant, unmodified, that you started with, what happens is this. Over on the left, where you see Texas 430, that's the inbred that this lab started with. See that it's kind of short and it's producing the way normal sorghum does. But look at all that other variation in the field. Above ground biomass, lots of yield changes, much higher yield changes, in some cases 70% increases in yield. Look at the vigor that you're seeing there. All that variation. We don't have this kind of variation to breed with in sorghum right now, but we do using this methodology. Now this is interesting, because it takes lots of genes to change yield, but in this case it only took one. More importantly, all those materials you see behind me in the field, they're non-transgenic. That transgene went in, made its change, came back out. So now, this is a new take on transgenic, isn't it? It's a new take on GM. Sure, we use GM technologies, but what we would sell in that sorghum is non-GM, but much improved. Of course, we can see very large changes in sorghum, but the interesting thing is we can in soybean as well. Look at the soybean on the left, it's modified. Look at the soybean on the right, that's what you started with. And these kind of changes happen in tomato. Look at this tomato on the right, and what you're looking at in the middle there is a graph of inflorescence and fruit set on that that's modified versus that that's not. No genetic change has gone on here that we know of, just the changes that occur non-genetically. Epigenetically. Now, why is this important? Because it gives us an opportunity, perhaps, to think about how we might actually make agricultural changes using transgene technologies that might, in fact, be acceptable to just the average consumer, okay? And because it's addressing the problems you need to think about for the next generation. What I want to point out here is basically in the red, you're seeing these modified plants and what they do for yield over time versus the unmodified. And you can see there's a point in time where you see quite a dramatic change. On the panel to the right, you're seeing that the next generation, it continues to occur. This is different than hybrids. Hybrids only last one generation. This lasts 5 generations. This now gets interesting. Maybe transgenics aren't so bad after all when you start talking about tomatoes producing on the right relative to the original tomato that produced on the left. This is a very interesting scenario, very new territory for the plant breeding community. And these are some of the new technologies that are coming with all those traits that I just described to you. Many of those traits I described to you are being developed on this campus alone. I haven't even told you what Illinois is doing, what Iowa's doing, what North Carolina's doing, okay? There's a lot of potential here. And we don't want to throw out all that potential without at least thinking about it first. This is the problem. We're going to throw out the baby with the bathwater before we find out what really can be done with transgenic technologies. If we could, in fact, double yields with simple changes like I described to you. There are other simple changes where we now have technologies where you can introduce a transgene that will go in, make one simple change to a specific gene sequence as you design it. Then that transgene is taken back out, but that change remains. Non-transgenic in the end, but beautifully precise changes that we need to make us more precise in our plant breeding efforts. These are really exciting technologies, all of them being practiced on this campus, all of them being practiced throughout the Midwest, and all of them a key part of what our solutions will be in the future. I want to leave you with a concept, with an idea, and maybe you'll even think about going to that website and getting a copy of this report. 150 plant scientists got together last year and got together in a room and sat down and hashed out what are going to be the key areas that we have to attack over the next 10 years. I mean, let's really get serious here because money is tight. After that meeting was held, 17 scientists got together, selected to represent the community, and hashed out how to put together a report to Congress that would lay out the next 10 years of plant science research that must be done to address the biggest challenges we have in agriculture. So I would encourage you to read that report. It's now publicly available at that website, or you can just copy down the title and it'll take you right to it with a Google search. But this report is, I think, important reading for giving you an understanding of the perspective that scientists have on all of the challenges that we face and why it is that we think that GM, along with other technologies, will have to be a part of this solution. Until we get past monoculture, until we can figure out a way to reduce the rate of population growth on this earth, we're going to need every tool we can find. So with that, I thank you very much, and I would be happy to address any of your questions. Thank you very much.
As is our custom, we'll now open the floor for some questions for Dr. Mackenzie to her lecture. And we have a number of people that are joining us online. This is also web streamed online, and we have the opportunity for some of those questions as well. So there are mics on both the lower level. Jill Brown has a mic here on the lower level, and I think Mary Garbetz upstairs in the back in the upper deck has a mic as well. So if you have a question, please get their attention, and we'll take some questions from the floor.
Professor Mackenzie, this is the second time I listen to your lecture, and every time I'm amazed about the way you perform up there and the information you provide to the public.
Well, thank you. I wish you would write the evaluations for my class then, because they don't say that.
I'll be happy to. My question is regarding some of the statements that you put in during your talk. One of them was no health issues detected yet. And being said that, and you mentioned about the several things that are not being addressed in the past. And I wonder, with this component of no health issues detected yet, what would be the control we have on environmental components affecting the responses on health issues on the public, or the responses of the farming or the products that you just showed? What would be the impact of environmental components, such as climate change, such as reduced or increased extreme events? How we handle those in terms of the approach that you showed us today?
So I don't mean to suggest that GM or transgenic technologies is a solution for what we're going to see in our challenges to crop production. That's really an oversimplification. Genetic modification using transgenes is, in my view, really at its infancy with regard to the kinds of traits that we're going to need to think about to address climate change. But my point is that we cannot get distracted with all of the barrage of misinformation that comes out about GM. We can't be distracted from what the real issues are associated with crop production in the future, because we must all be at the table. You know, we must all put our heads together if we're going to try to figure out how to grow crops with less irrigation, how to grow crops absent phosphate, how to grow crops that are going to be in a much more sustainable way, agriculturally speaking. These aren't simple questions, and they're not going to have simple answers. But the difficulty is that all of our attention seems to be drawn to all of the hyperbole associated with GM. And I want to convince you that it is, in fact, hyperbole. In fact, we can get beyond this, and we need to get beyond it in order to start addressing what really ails us. And that is an intensive monoculture system that is not going to be sustainable. And the Monsantos of the world know that won't be sustainable as well. And that is why you're seeing them coming out with traits like drought tolerance. Everybody gets it. We have to start thinking very carefully about what will gain yield, what will sustain yield on basically a diminishing agricultural and a decaying agricultural infrastructure. So we won't have control over climate, but we can learn to adjust. Mother Nature is remarkable in the way that those adjustments happen. And we can actually expedite some of that adjustment using some of these technologies. I just really aim to get us off this discussion and onto the discussions that are really the more substantive for the future.
I think there's a question in the back.
My question relates to where are they now? And one is Virginia Tech for a time was working on reducing the nicotine content of tobacco because the plant is so profuse in producing vegetation and using transgenic methods, but I haven't heard anything about that. And the other one is what has transpired with incorporating omega-3 fatty acids into the metabolism in the meat animals? Is that going forward?
Well, I can't speak to the meat question, but certainly in terms of tobacco being an absolutely wonderful biological resource, I don't know the levels of nicotine, but I do know that tobacco needs to remain a key model system. And in fact, in that decadal plan that I was describing to you, tobacco is one of the key model systems. Certainly for cell biology, it's a critical one.
On the omega-3 question that you asked, most of the work in that arena has focused on increasing or shifting the ratios of the fatty acids, not through transgenic technology, but through selection. Mary up top.
Dr. Mackenzie, we have a question from Ron Rosati. He says, can you summarize the valid criticisms of GMO crops, if any exist?
Yes. So in my personal view, I think that one of the criticisms I have for GM crops has been that there has been quite a lot of emphasis on profit, and there's good reason for that. You know, the regulatory system that is in place costs between $20 and $30 million to get a crop out. And as a consequence of that, it means that we have really narrowed our focus to only a very small number of traits for which any company willing to go out with that trait can get back the investment that they make in putting it out. And therefore, we have herbicide tolerance, we have insect resistance, but there is a whole host of things sitting on the shelves of most of the biotechnology companies that are absolutely wonderful traits for the consumer that we may not see. And it's because that $30 million won't be made up right away in those traits. We won't necessarily pay for what it costs to enhance nutrition or to create those novel traits. So that would be one of the criticisms. The other criticism I have is that, you know, it is common that when, you know, large companies are involved in deploying technology like this, you know, it's difficult to control it. And it's amazing to me how many acres are planted to GM crops because it is so successful. As a consequence, you know, we reap the problems. You know, the super bugs and well, they're not as much of a problem as the super weeds. They're our own fault. And any biologist knew that that was likely to be the outcome of recurrently using the same herbicide again and again. So I fault them for, you know, not having been better stewards in that regard. I don't know that the fault necessarily falls with big companies, but I fault the whole system, that intensive monoculture system, because this is an annoyance to any breeder who, you know, all breeders know better than that. But otherwise, in terms of the safety concerns, I don't see any yet.
Just to follow on to that question, one of the things that was really neat to see, you can't see from out there that we can see from here is the entire top deck of the auditorium today were students. And I think that's really, really a testament to both our students, but to the importance of this topic and how it's appealed to our students. And that last question actually came from our students watching at the Nebraska College of Technical Agriculture in Curtis, Nebraska, in western Nebraska. So other questions from the floor for Dr. Mackenzie? Right here in the center, Jill.
I have a couple of questions. One is, are the transgenic plants also patented as the genetic ones are? And if so, how would that affect the free market for agriculture? Because I think that that's a big criticism of Monsanto, is it owns the patent and the farmers can't keep their seed. And then the cross-contamination thereof of transgenetics as well. Does it cross-contaminate as the current genetic does? And then my other question is whether you're familiar with Dr. Don Huber and the research he's done on the broad-spectrum antibiotic of glyphosate.
Yes, so with regard to the issues associated with profit, you know, it is difficult to find a solution to this. We want to have new technologies like GM technologies regulated. And as a consequence, we put into place a very stringent, very intensive, very laborious regulatory hurdle or maze that anyone wanting to put out a new crop has to negotiate. This is why you don't see GM products coming directly out of universities. You can't believe the technology that's on this university alone, never mind throughout the Midwest. It would boggle your mind. It boggles mine how brilliant so many of my colleagues are. Those will not ever see the light of day unless they actually run through one of those companies. And that's because it costs $30 million to develop that crop, to get past the regulatory hurdles to do it. Well, I'm not arguing that those regulatory hurdles shouldn't be there, but then at the same time, you can't argue that a company shouldn't somehow recover that investment. $30 million for one product is a lot of money. Well, that's where that patent comes up, is it gives them that 20 years to try to recover the investment they've put in to making this go through. Now, you've got to understand, the 20 years of GM with all this hype isn't just the investment in developing the product, it's all the legal battles that you go through just to defend your product against all the people who believe that, oh my goodness, it made my dad sick. You know, you've got all of that going on. So basically, we've kind of created a no-win situation. If we're going to have GM, the patent rights are basically the only way a company can use this technology by virtue of the fact that the regulatory system is so difficult. What other system would there be? If I develop something wonderful tomorrow and I want to market it, how on earth will I get that to market? The legal hurdles are much more than any University of Nebraska could even begin to contemplate. So, you know, there's a no-win there. I would hope that over time, as we get to the place where we trust the technologies more, and perhaps the regulatory issues will be a little bit more defined and less prohibitive, then you will see an adjustment of all of that proprietary patent issue. But otherwise, you simply have a, how do I pay for the product? I don't see any other way. So, you know, you can argue it, but it's the American way. You know, for all the products, the idea that it's a plant product with a new transgene is really no different than, you know, basically we patent our varieties that are non-transgenic as well. We just call it something different. You know, that has gone on for many, many years, and it is only so that a company can recover all of their costs of development. Now, with regard to your second question, which was, Dr. Huber. You know, I worked for 11 years at Purdue University, and Dr. Huber was my colleague. Dr. Huber was a plant pathologist, a very fine plant pathologist. But Dr. Huber was not a molecular biologist and never did transgenic work. And so it's difficult to rationalize, for any scientist to rationalize, if you have data, and if your data are alarming enough to be alarming enough that the public should be notified, then basically it's your obligation, as Dr. Huber well knows, to present those data to the scientific community. The argument I have with Don is the same argument I have with Professor Seralini in Italy. If I have data, and my data are so troubling to me, why on earth would I not present them publicly to the scientific community? The scientific community is not biased in this way. What bias you see in the scientific community comes from a long number of experiments and many, many years of investigation. So any bias that the scientific community feels is just a fairly solid grounding in this science because we've done it for so long. But if I could come to this community and present solid, well-founded data, where statistically I've demonstrated this, where I've shown it to be reproducible, and it's showing that there are real serious health effects associated with the technology, you better believe all my colleagues would want to read it. And they'd want to know my reputation, and they would come back and they would grill me. And I better have my story hold up, because I'm basically coming up against the entire scientific literature base. But the fact is that they would listen to me. And if I put that scientific evidence out there and it held water, they would all basically accept that. That's the way science works. Science is not about your opinion or mine, it's about the data. And Dr. Huber knows that. He very well knows that. So the fact that he has not put forward those data, as much as I care very much for him as my colleague, I feel that was his obligation. And until he puts forward those data, I don't feel comfortable supporting his contentions, nor does the scientific community. This is not about bias. It's about well-founded data. Same as Serafini. I've never met him before. I'm sure he's a wonderful, wonderful man. But the fact is he knows full well he's obliged to provide primary data to the scientific community. This is important to us. We need those data. We need to evaluate those data. Otherwise, it's sensationalism.
We'll take one more question to bring us to a close. Mary, you have one up front, up top.
I'm interested in the increased production that you showed, the tomato plants. Mother Nature doesn't give anything for free. Any idea of what demands on the resources to sustain or realize that production, like in nutrients, water, and all the things that plants need to grow that way?
That's a possibility, and though that information's not yet available, this is very new technology. The question that usually comes with regard to Mother Nature never gives anything for free is the idea that if they're putting all their energy into that yield, what happens if they get hit with a drought? Or what happens if they get hit with a disease? Well, they basically tumble more readily. And the information isn't completely there yet either, but so far, that crop of sorghum was planted right out in Nebraska here in 2012. So it has seen its share of drought. And although everybody took a tumble, it actually outperformed the unmodified as well there. So, things are promising. This isn't something that's going to be out on the market anytime soon. But, it just gives us hope. My only point in pointing it out here is it gives us hope that there could be some compromise. Yes, transgenic technology, but basically non-transgenic product, that could work. So I think that that's an opportunity that maybe scientists could grab onto and pursue with a little more vigor. Yeah.
Well, beg your patience for just a minute before we make our presentation to Sally for her lecture. We do want to announce to you the next Heuermann lecture, which will be on Tuesday, November the 5th. It will be at the Cornhusker Hotel downtown, and it will feature U.S. Secretary of Agriculture Tom Vilsack, who will come to us and speak jointly to the National Rural Futures Conference and the Heuermann Lecture with the topic, Rural America, New Markets, New Understanding, Unlimited Opportunities. So that's Tuesday, November 5th, 2:30 in the afternoon at the Cornhusker Hotel downtown, U.S. Secretary of Agriculture Tom Vilsack. Please join me in thanking Dr. Mackenzie for a wonderful lecture today. Very stimulating.